All right, this is the start of a new section, this section on population genetics. Here we go. And it's the third out of 6 sections in the module ZOO3649. So in this section is where third year really starts, okay? This is where we are making a complete break from all of the very elementary stuff you learned in first and second year. Now, this is the more advanced stuff, and we're going to be talking about populations and the genes or alleles in the populations. And when we talk about those two things together, we're talking about population genetics. So what are the things that affect the genes in a population, or the alleles in a population? What are the things that affect it? Well, luckily, there are only 4 things that affect that, okay? And those 4 things, as you know, are the forces of evolution: mutation, selection, genetic drift, and migration. In this part of the module, we're going to be unpacking those in more detail. And we're also going to do a lot of simulations. We're going to do a lot of experiments, especially during the 3 practical sessions that you have to come for face-to-face. We're going to be doing a lot of experiments on the computer to see with our own eyes what these forces are doing to the genetics of populations. How did these forces control what happens to the genes in a population? Okay? And that is what the basis of population genetics is all about. So if we want to go one step back from here, we want to say, well, remember, back in the days of Darwin and Mendel, where we left off the last section, we didn't know too much about the forces of evolution, right? Those guys didn't know too much. In fact, Darwin was the first one to come up with the force of selection, right? Natural selection and sexual selection. And it was only later that other ideas like genetic drift and so on came about. So we're going to be talking about these ideas, and we're going to actually deal with, in this particular lecture, about how the 2 main ideas that I've told you about in the last section, become synthesized together in what is now the basis of population genetics. And this is what we call the modern synthesis. And whose 2 ideas are we synthesizing? What is a synthesis? Come on, right? What is a synthesis? Synthesis is putting things together, synthesizing, weaving them in together. Okay, making one thing a part of another thing, bringing things together. So the modern synthesis is basically, we call it modern, even though it happened a long time ago. At the time it happened, it was modern. And it's the synthesis of 2 big ideas. And that's synthesizing the ideas of Darwin with the idea of Mendel, right? The idea of natural selection with the idea of inheritance. So Mendel's idea was that the genes are the means, or the particles, as he said, right? Genes are the means through which information is inherited. So Mendel figured out how the information is inherited, and they pass through the germ cells. Okay? What was Darwin's idea? Darwin's idea was that if an individual's genes give it an advantage, it is more likely to survive and pass on those genes to the next generation. And that means increase its fitness. If you pass your genes to the next generation, you have a high fitness. So you see here, both ideas have a common thread. And you know what that is? It's the gene, right? So how do we synthesize these ideas? Because they're both dealing with the same thing, okay? So the modern synthesis is basically the central idea that brought these 2 ideas together, and is the basis of our modern evolutionary understanding. So most things we know about evolution, the nuts and bolts, came from the modern synthesis. And that's why you're being taught about the modern synthesis today. Give me a moment here. I just want to turn my sound down. Thank you. Right. Okay. So we are synthesizing natural selection with inheritance. And this is where the whole idea of populations comes in. Okay? Remember, this section is called population genetics. Why population genetics? Why not something else genetics? This is why. Natural selection acts on an individual, right? Thanks to its fitness. So an individual is a high fitness, they're going to the next, they're passing their genes to the next generation, their kids. Low fitness, you're not going to make it. You're not going to survive the struggle for existence. Maybe, or if you survive the struggle for existence, you're not going to survive the struggle for reproduction. Remember, that's all about sexual selection, the reproduction part. So, but either way, it's about your fitness, right? Natural selection operates on the individual. Natural selection cannot say the population is not fit. Natural selection doesn't care about the population. Natural selection only cares about the individual. You fit, you make it, you reproduce, you have kids. You're not fit, you either die or you live, but you don't have kids. Okay? But it's acting on an individual basis. Okay? It determines whether the individual survives long enough to mate and pass on its alleles. Okay? So an individual cannot evolve. Okay? If an individual could evolve, it would be Lamarckism, right? Where you evolve during the course of your life and give your kids an acquired trait. You see, that doesn't happen. We know that doesn't happen. Okay? We know that doesn't happen. You can't evolve. Your mind can evolve. You know, you can learn many things as you get older. You can become wise like an old go-go. But when you die, that wisdom is dying with you. Okay? A kid needs to learn all that stuff. Again, he doesn't just get it like that because you had it and you have a kid. Doesn't get all the thing in the brains in your head, right? Doesn't get all your wisdom. Has to develop it again. So you can't give the things that you acquire, no? So all we can do is live. We can be born. We can live. We can pass on our genes to our kids, and then we can die. Those are the only three things that we can do as individuals. Okay? That's it. That's the whole meaning of life. That is why we are here. All right? So you can see that an individual cannot actually evolve, right? Not in the sense of heritable evolution, right? You can change, you can become different during your life, but you can't evolve because you can't give those changes to the next generation. All right. So what is evolving then? If the individual doesn't evolve, we can't measure evolution in an individual, right? We can't. What is evolving? It is the population that the individual is a part of. That population is evolving from one generation to the next generation. Okay? Because in the individuals inside that population, they are struggling, right? According to Darwin, they're struggling for existence. They're struggling to find a mate. They're struggling to pass their genes on to the next generation. Okay? That's the individuals are struggling. Selection nails them one by one. And what happens to the population? Okay? Not all the individuals in generation 1 will have kids in generation 2, right? So some of them won't contribute. That means the alleles or the genes in generation 1 will not be identical to the genes in generation 2, because some of those genes or alleles will not be passed on because those guys were not fit. They didn't make it, right? So generation 1 will not have the same alleles as generation 2. Okay? And that is how we measure evolution. The change in the frequency of alleles from one generation to the next. So that is how we're going to define evolution in this module. There are many ways to define evolution. Okay? And we will go through a couple of them. And you can use any one of them if you're asked a question in a test or an exam. It doesn't matter which one you use. But I like the simple explanation that evolution is the total of the genetically inherited changes in the individuals who are members of a population's gene pool. So basically, the amount of change in a population's gene pool from one generation to the next. If there are changes, there's evolution. If there are no changes between one generation and the next, then there's no evolution. It's very simple to measure evolution. You just look at the genes and their frequencies. All right. So it's not something that is just a theory. It can be measured. And when you can measure it, it becomes real. Okay. So that brings us to gene pools. And that brings us to allele frequencies. Okay. You've heard about this in first year. You've heard about this. And you got a lot of it in second year 2544. Okay. So the gene pool is all the alleles in a population. Okay. You take one population, every individual with an allele in that population, all the alleles put together, that's the gene pool. Okay? All the alleles, not the individuals, the alleles. And in a diploid population, each individual will have how many alleles? 2, right? So if there are if it's a diploid population, the population size is say 20, what is the size of the gene pool? 40, right? The gene pool is 40. Because there are 20 individuals, each one has 2 alleles. So the gene pool is 40. All right. The gene pool is always double. In a diploid organism, the gene pool is always double the number of individuals. Right. So let us deal in this example, and you've seen this example before. So I'm not gonna take forever like I did in second year. Okay. I'm not gonna take forever. And besides which, even if I do it fast, you can watch it again. So there's no problem here. So let us deal with one gene. Okay, here is our gene pool inside this circle. Inside this circle is our population. All right, and you can see each individual is a circle. Well, it's not a circle; it's an ellipse, right? It looks like a rugby ball shape. Right, there it is. Right, that's an individual. That's an individual. That's an individual. That's an individual. And now you see the colors. Right, what are the different colors? The colors are the 2 different alleles. There are only 2 colors. There's only 2 halves. Right. Now look at these alleles. How many alleles are there in the pool? There are 3 alleles. There is the green allele, there's the blue allele, and there's the red allele. So now, so as not to be confused, how can you get an individual with 2 colors? It means what? It means that individual has 2 different alleles, and therefore it is what? It's a heterozygote. Okay? This guy here, where my mouse is, this is a heterozygote. Look at this one. This is also a heterozygote here. Okay? So and then what would this be? This guy has got the same allele from both parents. So he's a homozygote. Here's another homozygote for the green allele. Here's a homozygote for the blue allele. Okay? Each oval here is an individual. And how many individuals are in this population? You have to count them up. And then the gene pool is that number times 2, because the gene pool is the number of alleles, not the number of individuals. Okay, so when you are working out allele frequency, you must always divide by the gene pool and not the number of individuals. That's a big mistake everybody makes in these problems. You got a lot of these problems in your practical guide. Okay, don't make this mistake. You will be marked wrong, and you will make the mistake also in tests. Don't. When you are asked for an allele frequency, you divide by the gene pool. That means all the alleles. On the other hand, when you are asked for the genotype frequency, what's a genotype? For example, what is the frequency of the heterozygotes? That means you have to count all the heterozygotes in this population, for example, and divide by what? Here is where the smart people don't mess up and the silly people do mess up. You don't divide by the gene pool. You divide by the number of individuals, because each individual has one genotype, right? A heterozygote is one individual. A homozygote is also one individual. So if I want to say what is the frequency of heterozygotes in this population, you will count 1, 2, 3, 4, 5, 6, and so on, all the heterozygotes, and you will not divide by 2 times the number of individuals, the gene pool. No, you will divide by the number of individuals only. Okay, so genotype frequency you divide by individuals. Allele frequency you divide by the gene pool. Please don't forget. Right, so how do we now calculate the frequency of each allele? Remember, allele frequency you divide by gene pool. Right, so the frequency of the green, Now you have to count up the green alleles and divide by the gene pool. So what is it going to be? Remember, that's one allele. How many alleles is this? This is 2 alleles, right? You have to count that as 2 because he's got one from mom and the other same one from dad. Okay, so what's the frequency of the green? It's 12. If you count them, there's 12. And the gene pool, there are how many? There are 30 circles here. That means there are 60 alleles. So the gene pool is 60. And that means 1 over 5. 12 over 60, 1 over 5 is the frequency of the green allele, or 0.2. Right, now, what is the frequency of blue? Go on. That means you have to count all the blue alleles, 1, 2, 3, 4, 5, etc. And divide it by what? Again, 60. And that is 18. If you counted it, you will find 18. And that is 18 over 60, which is 3 over 10, 0.3. Now, what is the frequency of the red allele? The last allele. There's only 3. And here's a trick, right? A trick you already know, but I'm reminding you again. The trick is that if you have 3 alleles in a population, and I've given you the frequency of 2, you don't have to calculate the frequency of the last one, because what? All the frequencies have to add up to 1, okay? So you don't have to do the last one. You can add up the frequencies you have, and then what? Minus from 1. So what do you get? What do we have here? We have 0.2, okay? We have to add that to 0.3, right? That's 0.5 together. Minus from 1 is equal to 0.5 subtracted from 1 is 0.5 itself, right? So we know, without actually making any calculation, that the frequency of the red allele is going to be 0.5. Because 0.5 plus 0.3 plus 0.2 is equal to 1. Okay? You're not going to get 3 alleles in most of the problems in your practical guide and your tests. You're going to get 2 alleles. Easy. And you know, whether it's allele frequency or whether it's genotype frequency, if you're given 1 allele frequency, you can work out the other because it's 1 minus. Okay? If you're given 2 genotype frequencies, remember there are 3 genotypes, right? There's the 2 homozygotes and the heterozygote. Whereas in the problems here, we're only going to have, in your problems in the practical guide, we're only going to have 2 alleles, right? Allele frequency is easy. It's just the one you know minus from 1, and you get the other one. Genotype frequency, there are 3, right? So if I say I give you the 2 homozygote, the frequency of the homozygote dominant, the homozygote recessive, how do you work out the frequency of the heterozygotes? You just add the 2 and minus from 1. Okay? So very simple. And if you actually did it the hard way, we knew it was going to be 0.5 here. If you did it the hard way, what would you do? You would have counted 30 alleles and divided by 60 and got 0.5. Okay? The sum must always add to 1. All right. Genotypes must always add to 1. Alleles must always, frequencies must always add to 1. Now we are going to actually work a problem example. I'm going to go through this quickly because you have it. Firstly, you have it explained very well in your 2544 lectures, which you've downloaded. So go and look at your 2544 lecture if you want it slow, because I'm going to go through a bit quickly here because this is 3rd year. This is high school. I'm not going to waste 5 minutes in 3rd year doing high school stuff. Okay, you should know this already. We're going to go very quickly. So I want to know what are the allele frequencies of big A and small a. There are 2 alleles in this population, big A and small a. There are 3 genotypes, big A, heterozygotes, and small a, right? But we're not talking about the genotypes. We want to know the allele frequency. That means the frequency of big A and the frequency of small a. So that means we have to collect, right? We have to collect all those, all the big A alleles and all the small a alleles together. All right? So how are we going to do that? So let's see. I want to know the allele frequencies. Often it's useful to know what the genotype frequencies are. Okay? And in this case, you're given the genotypes, the 3 genotypes. You're given exactly how many in each genotype. Okay? And for example, if this was a problem for lethal alleles, you will be only given these two genotypes here. You will not be given the frequency of this, because why? This one should be dead. It's 0. Okay? And you say, Prof, you didn't give us the recessive. I did. I said it was a lethal allele. That means it's 0. Go and work out the problems. You'll see they're easy to work out when you figure that out. But coming back to this one now. Here are the 3 genotypes. This one is not lethal. There are small a, small e is not lethal. It gives you a white color in this particular organism here, right? In this flower, in this plant. So what are the genotype frequencies? First, let's work out genotype frequencies. So that means how many big A's do we have? We have, we have 320. And to work out the genotype frequency, we do what? Divide by number of individuals. Okay? So it's 320 divided by what? How do you know the number of individuals? You have to add all these 3 numbers up. Then you know the total. Then you divide by that total, which is 500, and you get your 0.64 for your allele frequency, genotype frequencies of big A, and you can do the same, exactly the same for the genotype frequencies for the heterozygotes, and exactly the same for the genotype frequencies of the homozygote recessive. Okay, but now we didn't get asked for genotype frequencies. Okay, if you got asked for genotype frequencies, that's how you do it. Remember, you divide it by the number of individuals, which is 500. Now, in this second step here, we want to work out the allele frequency of A and B, so P and Q. Okay, big A allele frequency is P, small a allele frequency is Q. All right, so now let's do it. So we got to collect all the big A alleles, right? We are forgetting about genotypes. We're collecting just the alleles. So this big A, this flower, we know there are 320 big A's. That means how many actual alleles are there, big A alleles are there in this 320 individuals? Each one has 2 big A alleles, right? So that means it's 320 times 2 gives you 640. So these guys here have 640 copies of the big A allele, okay? Because there are 320 individuals, each one has 2 copies, so therefore we will have 640. But, ladies and gentlemen, that is not the only big A in this population. Where else do we have big A's? Of course in the heterozygote, right? This big A, small a. You can't ignore that one. There's big A there, and I want the frequency of big A. So you have to count these. But you don't count them the same way you counted the heterozygote dominant, the homozygous dominant, right? Homozygous dominant, we simply just took 320 individuals times 2, 640. We're done. We've taken all the big A's from this genotype. We want to take all the big A's out of this genotype. How many big A's are there in this genotype? There are 160 individuals. Each one has 2 alleles. But in the heterozygote, only one of the 2 alleles is the big A. The other one is the small a. So you don't have to multiply by 2. You just take the 160. No? Right. So here, you've taken the 160 of the big A, and you add it to the 640 of the big A from here. And so together, you've collected all the big A's. Okay? You see how you've collected them. You took them from the homozygote dominant, but you also have to take half of the heterozygotes. So you collect all the big A's, not just the ones in the dominant phenotype, in the homozygous dominant phenotype, right? So it's 640, so that's 320 times 2 is 640, plus 160. We don't times by 2 because why? This is the other 160 is small a. It has to go to this side when we're collecting small a's. But now we're collecting big A's. We go 640 plus 160 is equal to 800. Now we divide by the gene pool, not the individuals. And the gene pool is what? Remember, the individuals, if you added them up, they are 500. That means they are 1,000. Okay, the gene pool is 1,000. The individuals are 500, and the diploid organism, the gene pool will always be 1,000. All right, 2 times the number of individuals. So you divide it by 800 by 1,000, you get 0.8. Okay, so remember I said to you, you don't actually have to work out the other one if you worked out the first one, right? Because it's just 1 minus. So the allele frequency of small a should be 0.2. But if you do that, if you just work out 1 and minus from 1, then you don't have another check to see if your calculation is correct. What if you made a mistake when you were adding or something, and then you got the wrong answer for A, as for big A, for P? Yeah, frequency of big A, 0.8. What if you got the wrong answer there because you made a mistake while adding? Then you take that one and minus from 1. That means you get the wrong answer for the Q as well, for small a. Right? So it's always better, even though you can simply minus from 1, it's always better to work it out. No? So let's work it out. So we minus from 1, 0.8, we got 0.2. But let's work it out so that we know we are sure about both. And then we've double checked. When I ask you to go back and double check your answers, this is what I'm expecting you to do. Do the calculations again. Okay? Don't leave the test early and then you get less than 50%. That's nonsense. You have time, you sit there, you do the calculations again, you get a pass mark, you maybe even get a distinction. Right, so how do we do this, guys? How do we collect up now all the small a's? We've collected the big ones, how do we collect the small ones? Where are the small ones? Here they are. Small a, there's 20 individuals with small a. So how many small a's if we have to collect them all up here? It's 20 times 2, right? There are 40 small a's among these 20 guys here because both of them have 2 copies of small a. But that's not the only small a we have in this population. Where are the other small a's? They are hiding where? Here in the heterozygote, right? Remember, you took 160 of the big a's, you put it there, you've figured out the big A frequency. But now you need to work out the small a frequency. So how many of them? There are going to be also 160 small a's here. So you take those 160 small a's, you add it to what? To your 40 from here, right? 60 plus 40 is 200. 200 divided by the gene pool, which is 1,000, is 0.2. Yes, hallelujah, our numbers make sense. So you can minus from 1 to get 0.2. But if you want to double check that you didn't make a mistake, then you work it out again and you still get minus 0.2, you know you got the right answer. Okay, please, if you got any problems with this, watch this video again and again and again until you get it, because a lot of your problems in your practical guide are like this. They're asking you for this data. They're asking you problems about these kinds of things. Okay, so please go through this example again by yourselves and make sure, please make sure that you know how to work out genotype frequencies and how to work out allele frequencies. Okay, so basically, if the allele frequencies in the population, now we know how to work them out, right? We can measure them and we can figure out what is the allele frequency, as you can do now as well, right? So now we take a population in generation 1, we figure out the allele frequencies. Then we take a population, we wait for them to mate and have kids and so on. Now we go to the second generation. We work out our allele frequencies of the second generation. And now we can compare the 2 allele frequencies. Generation 1 and generation 2. And if they are different, what does that mean? It means the population is evolving. Okay? It means the population is evolving. All right, let's do an example. Here is a trait determined by 2 alleles, big B, small b. Frequency of the alleles in generation 1 was 0.92 and 0.08. Remember, if you add these 2 up, there must always be 1, right? Allele frequencies. But now, these guys had offsprings, okay? And the offspring are have their own gene pool. And now we've come and we've measured the very same alleles in the offsprings. What do we get? We get 0.9 and 0.1. So that means this one, big B, went down from this generation to this generation. Big B went down in frequency by 0.02. And small b went up in frequency by 0.02. So the frequency is not the same from this parent's generation compared to the offspring generation. That population has evolved. Okay, that's how we measure evolution. We can safely say evolution has occurred between those 2 generations. It's so simple. And everyone thinks evolution is hard. It's not. That's all there is to it. Okay? The entire population's gene pool has evolved towards a higher frequency for the small b allele. Okay, so that's how we defined evolution in the first place, or quite simply, any change in the frequency of alleles in the gene pool of a population. Okay, when the frequency of the alleles change from one generation to the next, you've got evolution. All right, so just to give you an idea in this last slide of how a population evolves now from one generation to the next. So look at this diagram here, okay? Look where it says past and where it says present. Present is now. Now we've got these two species, species A and species B. But in the past, they never used to be two species, right? In the past, there used to be one species here during the time of the common ancestor of these two. Okay? So, here you have time, and at this time here, all the alleles look like squares. And at this time here, you have a circle coming, evolving. And at this time here, you have another color circle evolving. And at this time, as you're going down from past to present, the alleles are changing. You see the population is evolving. It's becoming different. The alleles are changing. And then in this area here in the middle, right, there are some alleles that are in species A, like these square alleles. They are there they are. But then there are other alleles like these round ones which are only in species B. But there they are. Both the square and the round are around during the time of common ancestor. Okay, so that is how new species evolve. So from one generation, you have a small change in allele frequency to the next generation. From that generation, then you have the next generation, and again a small change, and again a small change, and again a small change. And I put this diagram here to show you from one generation which is there to another which is there. The change is small. But if you look at it over a long period of time, like this graph is doing, then you can see how a population over small changes from one generation to the next can acquire big changes over a big period of time. That was one of the things Darwin needed to know before he could come up with his natural selection theory. You know this, I explained it to you. He had a friend called Charles Lyell who was a geologist. And Lyell figured out you need a long time. The Earth is a very old place. It's not 4,000 years, it's billions of years old, okay? So the Earth is an old place. And so evolution has taken a long time to happen. Okay, I'm going to leave that with you, and we will continue our next lecture, lecture 18.